Welcome, everyone. So today we have a talk by Paul Macklin, who is from Indiana University, and he'll be telling us a little bit about, well, the title of the talk is Mapping Knowledge to Understand Dynamics in Human Tissues. As part of that, you might hear a little bit about ontologies and perhaps a little bit about the HubMap project and the involvement of Paul with that project. And I'll just let Paul take it away. Thank you, Paul, for joining us.
Well, thanks, Raphael, and I really appreciate all you and all of your group for inviting me to share some of this with you today. Let's see if I can get the screen share working here. So first of all, I want to kind of begin by acknowledging funding for some of this work over the past several years. Some of the immune modeling that we'll be showing you was particularly funded by the Jane Koskinas Ted Giovannis Foundation for Health and Policy. A lot of the cancer modeling work has been funded by the NCI over the years, including a supplemental, an administrative supplement. Some of the cloud-hosted work that we may be showing has been funded by the National Science Foundation, and some of our more recent work in cancer patient digital twins has been funded by the NCI and the DOE. And then other recent immunologic work has been funded by the DOE and Defense Threat Reduction Agency. And then, of course, HubMap is funded by the NIH Common Fund, and we're very delighted to work with them. Let's see. So first, I want to give, kind of a quick primer on HubMap and to kind of contextualize some of the work I'll be sharing. I'm not going to really spend the majority of the talk talking about HubMap specifically, because really the right experts to talk about that is probably Kade Berner. But I want to give a special thanks to Kade as a PI of our component of the HubMap, and thank her for sharing some of these slides to help share today. And to kind of give an idea, first of all, an overview of what the HubMap is, it stands for the Human Biomolecular Atlas Program. And its idea is that it wants to catalyze a formation of really a comprehensive whole body atlas of cells and tissues, really going down to the molecular level of molecular characterization of cells, spatial annotation of cells in different sites throughout the body, and forming this in a coherent, unified way that you can search and find a tissue, find properties about it, go and get information about the cells in that tissue, and then learn something with that. Of course, as part of this, it really has a lot of goals that kind of go along with that you want to, realizing that this kind of endeavor of whole body molecular mapping is really kind of aspirational. And it is driving, not only using technology, but driving technology. And so a great aspect of HubMap is that it has kind of forced and helped boost the development of next generation tools for mapping, and then for registering, spatially registering spatial tissue maps. It is also generating, some foundational 3D tissue maps at important key sites throughout the body. We'll talk a little bit more about that later. It's also creating infrastructure, an open data platform to allow people to insert data into this map, and of course, later on to access it and combine it and analyze it. And one other key role that HubMap has been playing is as, kind of a large federally driven mapping program developing this infrastructure. It has been trying to reach out to other related projects, particularly like the Allen Institute, who's done a lot of mapping, and Chan Zuckerberg has done a lot of mapping as well, to kind of say, that there are a lot of groups that are doing one or more specialized maps. And if there can be some unification of the data standards and representations and infrastructure, these could be kind of co-registered into a larger, more comprehensive map that any one group could do alone. So there's a wonderful opportunity to help coordinate work in this community. And then, of course, to support projects that demonstrate the value of the resources developed by the program. You kind of have that, Kevin Costner, baseball field in the middle of Iowa saying, build it and they will come. And so we really would need to make sure that once this infrastructure is built, that people really do come and demonstrate the utility of it to kind of drive further progress in this shared resource. Want to point out that HubMap is really, over the last several years, grown to be a beautiful national and even international effort with sites all over the place. So here we are, I don't know if you can see my mouse, we're in Indiana driving some of the core infrastructure, but there are mapping centers and infrastructure components spread all across the US, and as well as collaborations in the Netherlands and the United Kingdom to develop some of the key infrastructure and technologies for HubMap. And so in particular, there are a few different types of participants in this HubMap. really as a network of traditional, U-scale grants, like programmatic grants, and then contracts, OT2 type contracts that develop all the core technology. And these groups all work together into a broader network that is HubMap. And so particular, there are groups in these TTDs, or these transformative technology development groups, that are really here to accelerate development of key technologies that are identified as needed to perform the core functions of creating these atlases. Of course, at the center of it is these tissue mapping centers. And so you have the groups that really have high-end state-of-the-art microscopy, characterizing a variety of tissues, who really are going to be run by domain experts in particular tissues, performing the mapping, and ultimately generating the data that will become the maps. But raw data by themselves don't make an atlas. You really need, technology, in particular ontologies and data standardizations and data processing, to be able to take that raw data and turn it into something that really resembles an atlas. And so together, this group of technology development has been called HIVE. So it's the hub, H for the hub map, and the I is for the integration, visualization, and engagement. And so these are kind of like the core technologies that together enable going from just individual data in images to a comprehensive atlas, and to not just map it and register it together and to kind of coordinate it, but then also to make it searchable and usable. And so you put these components together. You have the tissue mapping. You have, as you do the tissue mapping, realize that you need to develop core technologies that then hooks into the TTDs, and then integrating it all together are members of the HIVE. And IU is one member of this HIVE. And so in the last several years, the tissue mapping centers have really been focused around several key organ sites, in particular mapping the bladder, the kidney and the ureters, colon, lung, lymph nodes, spleen, thymus, and vasculature. And the TTDs have been testing out new technologies in other sites like the lungs, breast, liver, and tonsils. And so these have been the key sites, so clearly this is not the whole body, but some important parts of it to kind of start forming that basis for that long-term vision of creating a whole body atlas. And so these have been the focus for the last several years. So IU in particular has been playing a role as a member of the HIVE. And so our role is to kind of create the coordinate system that helps to tie all this data together. So we call this the CCF, or the Common Coordinate Framework. And the idea is really that, when you have raw data coming off of any one of these tissue mapping centers, it's going to span a broad variety of scales. It could be genomics, it could be epigenomics, transcriptomics, proteomics, metabolomics, pretty much anything you can stick the word omics at the end of. There's going to be some kind of characterization of a tissue sample, some of it spatial, some of it not spatial. And what we need to do is to say, we need the context, we need the addressing, and the CCF allows you to kind of take this information that you've mapped and to kind of integrate it into a broader coordinate system that says, you're within this organ, you're close to these particular landmarks within that organs, which are going to be very organ specific. And then from there, you can kind of annotate all the different things that you've imaged. And so this is being accomplished at IU primarily through development of ontologies that are helping to unify the nomenclature and the relationships in coordination with the TMCs and the broader community of experts to make sure that the ontologies are really driven by domain experts. So sometimes they find that the ontologies that are out there already are expressive enough. Sometimes we find they're overexpressive, and it's a matter of kind of finding and picking and choosing the best pieces of the ontologies that are out there, and then filling in the pieces that are missing into something that's going to work across all the different organ sites and the technologies being used across the TMC. Other things that, of course, is that to facilitate gathering data that can be properly registered into this atlas, IU has been coordinating with other members of the HIVE and the broader HubMap to help develop protocols for specimen registration, saying that if you map some kind of a tissue, you need to capture that context of where it came from. Can you register it, say it's from this position within the kidney relative to these landmarks, so you can actually identify where that tissue came from? Because we know there's going to be variation in tissues as you go across an organ, even if they look like repeating units. And then to help facilitate that, because that can be a very painstaking process, IU has been working to help develop user interfaces to facilitate, both the registration process, then later on to say, now we have all this data, but it's not useful if you can't access it and find it and make use of it. It has to be fair, and in particular, it has to be findable. And so they have been developing interfaces to help facilitate searching through the data that have been mapped, to visualize them and to kind of help to that data exploration that's going to drive new discovery. So let's see here. So at the core of the common coordinate framework development has been creating something called ASCT plus B tables. We'll get to what that means in a moment. And I really want to point out that this work has been led in particular by Kadi Brunner, Griffin Weber, who is in your neighborhood there at Harvard, Mark Musen, and David Osumi Sutherland, who are both ontologists, and then Ellen Kordorkas, sorry, got a tongue twisted here at IU, who has really been really at the heart of a lot of this work and kind of playing the translator role between very different disciplines involved in HubMap. And so there are a lot of requirements to build a successful common coordinate framework. And in particular, it needs to be able to capture the major anatomical structures that you're going to encounter across the human body. It needs to be able to, at the very least, clearly identify the specific cell types identified throughout the different tissues in the body. Sometimes these are cell types that are found only in specific organ sites. And there needs to be basically some reproducibility to it. And so if two different groups say that they've identified a particular cell type, it's very hard to actually be sure that those two groups are actually talking about the same cell type, unless you also say that that cell type corresponds to some very particular biomarkers, so that we can all be sure they're on the same place and have the same definition of the cell types. Because every day comes out and there's going to be a new paper coming out that someone did, some massive UMAP, decomposition of scRNA-seq, and they've discovered 15 new cell types. Well, we can't really be sure if those are new cell types until you have a little bit more commonality and clear definition of how we're defining these cell types in terms of biomarkers. Now, these common coordinate frameworks and their ontologies need to be semantically explicit. In particular, they need to build upon existing ontologies and standards out there, such as Uberon, and they need to be spatially explicit. So they need to be able to not just record molecular properties, but also spatial properties like XYZ position, proximity to landmarks, and all of this. The other thing is that the body itself is kind of inherently hierarchical. And so that hierarchy itself is already kind of a coarse and increasingly refined coordinate system as you go down from level to level to level. You start off with a body and say, well, okay, I'm in a body, then I'm in a particular system. Now I'm within an organ. Within that organ, there might be one or more functional tissue units. These are kind of like the smallest organization of cells that perform a particular function in that tissue, like a nephron in the kidney, for example, or renal corpuscle. And then when you get within the FTU, you might find substructures like Bowman's capsule or glomerulus. And then if you get even farther in, then you find the things that we're more familiar with in terms of biology, things like cells or basement membrane in particular cells. And then, of course, you could always go farther down to get subcellular localizations. And so you need this hierarchical level of addressing to kind of go from the very broad to kind of zero and say, okay, I'm in an organ, I'm in a certain functional tissue unit that might be in some spatial position. And then where am I within that functional tissue unit, and what are the different cell types out there, and what are their positions, and what are their, of course, their states as measured by this multi-scale characterization. And so the ASCT plus B tables have been designed to kind of help facilitate and systemize the knowledge that's being gathered across the different sites. And so the AS stands for anatomic structures. And so we need to be able to develop on an organ by organ basis, create an ASCT plus B table that captures different things. The AS are the anatomical structures that can be found in that organ. The CT says that within those anatomic structures, what are the types of cells that can be found? And then the biomarkers then are how we know we're all talking about the same cell types, saying, the parietal epithelial cell, for example, in the Bowman's capsule is identified by CRB2, CLDN1, or in the glomerulus, a podocyte would be identified by an MPHS2, PODXL, MPHS1, and so forth and so forth. And so now we have kind of a very clear molecular definition of the different cell types, saying, we're all on the level playing field, and now we can all be sure we're talking about the same thing because we have the same definitions and the same terminology. And so putting these tables together, and the ontology kind of tells you the loud terms, but the table then kind of gives a little bit of additional structure to that. And so this has been developed really on an organ by organ basis. And so IU's role, in particular led by Kadi Berner, Jim Gee, and then Ellen, serving as kind of our local knowledge manager, has been leading a working group that's been taking monthly meetings to go through organ by organ to gather domain expertise and to lead the community towards consensus to say, let's put together our domain knowledge and gather it all together and define these clearly well-defined ACST plus B tables for over 24 tissue sites. And so IU has not been writing them themselves because that wouldn't be the right thing to do. It really critically depends upon interaction between HubMap and the broader community of domain experts. And so that's something where IU has really been able to play a role. And so if you'd like to learn more about these tables and the way they've been developed for over 24 tissue sites, I really encourage you to kind of reach out to Kadi and Ellen in particular, Mark Musin as well, who have a whole lot of beautiful information on this. And then there are some preprints available, so I can make sure to get you links to those preprints, as reference material to kind of follow up from today's seminar. And so, looking at these typical tables, this is on an organ by organ basis. And so there are quite a few of these, but they can get to be actually quite structured, right? So here, looking at the kidney is one of our typical examples. You might start off with a structural region. So here's an anatomic structure, and renal corpuscle. Then within it, you have, say, Bowman's parietal layer, and then Bowman's visceral layer, and a glomerular tuft. And then within these different subregions, we have a list of kind of known and allowed cell types that have been discovered by saRNA-seq. And those then feed into an ontology that records these, this hierarchical relationship of the structures and the types, and can also be mapped onto a 3D reference, basically artist rendition, but not just an artist rendition, but one that's been driven by actual samples and one that's been driven by consensus among domain experts say, we agree with this picture and it has the right cell types in the right places and it matches what we've seen to date. And so these 3D reference objects are not just beautiful illustrations, but actually represent a spatial version of consensus building. So the tables are kind of like the structure of the semantic terms, whereas the reference object 3D visualization kind of says, now how do we arrange that spatially to reflect what we think today is the consensus knowledge of that particular organ. So you can see how this becomes quite a big undertaking to try to herd the community organ by organ by organ towards some consensus. The good news is there are only finitely many organs, so eventually this will be done. And so IU has been prototyping this on the handful of organs that you saw, at the beginning of the slide that have been funded, and then reaching out to other partners to go beyond the HubMap to try to perform this process for other organs that HubMap itself hasn't been mapping, but we know the data are out there if we can just reconcile the ontologies between these different groups. And so, again, we kind of get to these reference visualizations, but they're not just pretty pictures and 3D renderings now, but they actually reflect, that domain expert consensus for the overall structures. And by coming up with these tables and these well-defined ontologies and relationships among the terms, this has been allowing the CCF to branch beyond, again, as we mentioned, branch beyond the HubMap data that have been measured in the last few years for this handful of organ sites, and to reach out to other consortia to see what they've mapped, and then to map them onto a broader, this broader whole body index. So by spending this time on really carefully identifying and developing and getting domain agreement for these ontologies has really allowed HubMap to grow beyond itself, to grow bigger than the sum of its parts by taking advantage of the broader community. And that gets back to that original goal we talked about in the earlier slide, where HubMap really wants to play this hub role of integrating atlases and data from a broad variety of sources and not just from the internal tissue mapping centers. So that's a lot about the ontologies development. I'm afraid I'm not going to get really deep into the ontology, technical information. I just want you to know that it's out there and is one of the key roles that IU has played as a member of the Hive. Other aspects of IU's role here has been to help integrate new samples into the CCF so that they become registered into the broader coordinate system. And so here again, we have these tables, they have all the allowed cell types to kind of say that once you map a tissue, you know what ought to be in that tissue based on where it came from, but then we also want to map it onto the landmarks in the reference system. And so here's this is kind of, I guess it's long enough after lunch that this is hopefully not too gross, but there are a lot of tissue samples coming off. So for example, Sanjay Jain has been mapping kidney tissues. And so you might start off with the tissue that's been kind of drained and cleaned up. You have scale bars next to it. And, this is right now, you've got some pretty coarse measurements here. And, you want to somehow map this onto reference coordinates. And so IU has been leading interfaces that actually give you kind of an interactive system saying that as you start chopping up this block, first we want to say, where do we actually find this thing in the original tissue? And so you can have this 3D interface that allows you to kind of move around your bounding voxel to say, here's where my block came from, here's the size and the dimensions that we measured. And then from there, you can kind of register that into the broader tissue. The other key role that IU has been playing has been to create user interfaces to explore the data that have already been gathered. So the beautiful thing about ontology is that it gives you this nice hierarchical, click, 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 and navigate your way down to where you want to go. So you can say, okay, I want to start with the body, move into the kidney. Okay, now there are two kidneys, so let's move into the right kidney or the left kidney. And then from there, you're in a specific kidney, and you might say, okay, what are the blocks that have been mapped here so far? Maybe we can click one of those and zoom in a bit more further, and finally get to the original tissue sample and layer on different aspects that have been labeled. And so now we have some spatially annotated data. We can really visualize that original raw sample and eventually, download that for using your own analysis. And so as this goes forward, we're going to be approaching, this point where we are past mapping and spatially annotating data, and we want to say, what can we actually do with it? And there are a lot of key questions that I think a proper, fully spatially annotated, map would allow you to answer. In particular, there are a lot of cell types that are all over the body, not just within them. Endothelial cells, immune cells, other key cell populations, are found all over the place. There are broad epithelial cells, but they're very specialized by different organs. How do they differ? How are they the same? What's unique? In particular, take, say, macrophages. Are there specializations within specific organs? I mean, is there such thing as a kidney-specific macrophage, or are they a little bit more generic? Are there key markers for these specializations? Other things you can look at, in particular, Griffin has been looking at this kind of information, saying, suppose that you robustly map the blood vessels that are and the topology of the blood vascular networks within these different functional tissue units. Well, some questions we can naturally ask is, we have, say, spatially annotated cell states. So we say, here's a cell, here's a position, here's its type, here's the saRNA-seq data, here's metabolomics data, here's its other transcriptomics data. How does that state vary with how close you are to a blood vessel, or how close you are to a membrane, or to other cell types? So there's a lot of spatial analysis to kind of say, how do different cell states vary and correlate based upon who they're close to and who they're interacting with? And that might be able to help you infer how different cell types are actually interacting within these very complex tissue structures. And of course, what we really want to know, right, is how the tissue architecture relates to cell state, and how that cell state feeds up to drive the function of a microarchitecture to become the functional tissue unit, and how the microvascular network topology may relate to the structure and function of an organ. So there are a lot of things we can learn from HubMap, but critically, at least right now, HubMap is missing some key details. In particular, it's based upon a lot of static imaging. You have a donor tissue, which we're of course very grateful for, but it's inherently static. You take a picture at one time and that's it. So there are a lot of questions out there about what are the dynamics of a functional tissue unit? How do the cell states determine and reflect the cell function and the tissue function? How does the specific architecture actually lead to that tissue function? And how is it maintained? I mean, one big question actually we found in just sitting down in chats with domain experts saying, suppose you've mapped out the glomerulus in the kidney. If I measure it today and measure it tomorrow in the same patient, will it look the same, or is there constant remodeling? How static are those podocytes? Do they just form an architecture at birth and then they're done? Or do they constantly grow and remodel? How are these tissues maintained? And in particular, how robust are they to a perturbation, say an illness or an injury or cancer intruding or an autoimmune a disease or inflammation? And so we need to get beyond just the static pictures of the tissue, which pretty much says, here is a tissue, here's who's in this tissue and where they are, and here's their state at one point in time. But now we need the dynamics, and which means we need to say, not only who is here in this tissue, but what do those things do? What do the different cells do? What are the rules of endothelial cells? What are the rules of macrophages? What are the rules of CD8 positive T cells? And so we need to capture that domain knowledge to say, now that we've mapped a tissue, we know what's there. Now, if you show that picture to biologists, they're already mining that domain knowledge that they have to say, aha, that's really cool that I have these 4 different cell types in close proximity. But that information is not automatically transmitted to other people reading that map because they need that domain knowledge context to read the map. So we need to capture that domain knowledge to make the map more useful. In essence, we need to perform knowledge mapping on top of just the raw structural mapping. And so what my lab is involved in particular within HubMap is trying to move beyond this static structure to the dynamics and the function. And so this is really, I hope as a mathematician, we can play a clear role as that mathematical and data modeling can help to frame these problems a little bit more precisely. That domain knowledge capture can really be thought of as trying to make small and simple and clear observations and hypotheses. For example, macrophages may increase secretion of pro-inflammatory factor like IL-6 in the presence of dead cells. Or CD8 positive T cells may be drawn chemotactically towards IL-6. Or CD8 positive T cells may release things like perforin and lytic granules when they come in contact with a recognized foreign agent. And then those things damage a cell, and then damaged cells tend to die. So you put all those things together, you start getting kind of the key functionality of an immune response. And overall, we have lots and lots of simple statements that really come down to some cell behavior is modulated based in response to some kind of an environmental signal. And so we want to be able to write down in some way these simple things that biologists know and learn, but for us who are in data modeling or in mathematical modeling, this is kind of that knowledge that's far more valuable than just pure data because it takes such experience and domain knowledge and insight to get to. And so if we have an appropriate data model, which of course is going to require a controlled vocabulary and a grammar, then the hope is that you can capture that systematically in a way that can be mapped directly into a mathematical representation to help systemize that knowledge and to kind of say, now we can start getting at the thought experiments, if you will, the computational thought experiments to say, we have these rules and we've known them for a long time, perhaps, or at least we think we do, but they're often contradictory. And we don't know how the body balances contradictory signals. And this is the role that mathematical modeling can play, is to start balancing these competing hypotheses to see how we may find the balance between pro-inflammation and anti-inflammation, for example, in homeostatic tissue, between proliferation and death, between adhesion and repulsion. There's always these forces that are kind of in opposition in tissues and in physics and in biology. And when they're in balance, we have working tissues in homeostasis, and we have cell turnover is working just right and differentiation working just right. But when you just tip the balance a little bit, you start moving towards a disease process, towards autoimmune diseases, towards cancer, towards some kind of a degradation process of a tissue. And so we want to be able to systemize this knowledge to understand how tissues work and how they're maintained and how we can read in information for something like the hub map and infer some function from that static snapshot. And so that's what I'd like to spend maybe the last half of this talk here today and kind of getting to our role in trying to extract the knowledge that goes along with the tissues to make those tissues dynamic. And so this kind of notion really suggests a particular form for the data and the mathematical models that, we need the cell types, which we've been mapping in HubMap, and can say we have a bunch of cells, we have a bunch of positions for them. So we know the population, but we also need the microenvironmental context. What are the diffusible substrates that are important in this tissue? What are the important signals? What's the structure of the non-diffusible elements like the extracellular matrix? What are the physical parameters for all these diffusible substrates and chemicals? Because the balance of those parameters is going to affect things like communication distances. And so we need to be able to capture and describe and emulate the dynamics of the tissue environment. So to capture the state means we need a data model. To capture the properties of it means we need a mathematical model so we can actually move to the dynamics. Then likewise, we need to be able to describe the cells. And HubMap so far has done a really great job of capturing the states and the identities. But now we need to mine the knowledge to say, given a cell identity, what is its behavior? What are the rules of that? And can we structure the data around the key behaviors in a way to kind of systemize the otherwise mind-boggling variety of biology? And so several years ago, we started a project called Multicell DS. We had already been involved in agent-based modeling of cells, in particular in cancer, for a long time. And we'll get back to what agent-based modeling is in a moment. But what we really realized is that there's a lot of kind of do-it-yourself work by every lab, saying, that every cell, let's say you're doing a model of a tissue, and some lab comes up with a model of epithelial cells, and then macrophages, endothelial cells. They might develop a beautiful model of angiogenesis and vascular flow. They write their data, they write their model, they simulate a few things, they save their data in their own proprietary format, and then they kind of go away. And then the next person comes along and they do the same thing. And there's a lack of compatibility between the two. And frankly, even just a lack of properly annotating what the hypotheses are driving the underlying mathematical model. What do you actually think is going on, and what are your assumptions that drive that particular model? And then on the flip side, once you write data, because we can't be sure we're writing about the same cell types, and we don't even have the same representation of the data coming out of the simulation models, how can we possibly develop unified tools for analyzing and comparing models together, and maybe feeding knowledge from one model into another, or stringing together pipelines and workflows of, say, data assimilation techniques into a simulation model, and then post-processing and analysis. And so what we need to do is kind of move beyond individual simulators and really think more about trying to describe kind of the fundamental processes that are really in the data. And we looked around, several years ago when we started this, it was like 2008 or 7 or something, 2010 maybe. What we found was that there were, where there'd been the greatest success was where the biology and the chemistry was best defined. In the omics spaces and the genomic spaces in particular, and then in mathematical modeling in systems biology markup language, annotating chemical reaction kinetics, because there was kind of a well-defined mathematical form. And so it was really easy to kind of come up with a standardized canonical representation. But then when we got to multicellular tissues and tissue engineering and tissue biology, all that was out the window, and there was just no unified way to represent tissues and tissue states. So that's where we decided to work. And so we want to come up with a markup language that described not necessarily the depths of the models, but we just want to say, can we characterize the key behaviors in these models, things that we all agree on, like cell cycling, cell death, motility, maybe secretion and diffusion of chemical factors. And you say, can we write down those things that we all agree on as inputs or biophysical characterizations of key processes, and say, well, what do we need to model, to characterize in a way that's, model agnostic. Say, what's the time scale? What's the cell velocity? What's the cell size? These are things that are fairly objective that individual models are going to map onto their own individual parameters. And so if we can kind of characterize these, then we might be able to create something that's a little bit more flexible, more amenable to physical characterization. And so Multicell DS came up with a handful of data types through a couple of years of coalition meetings, saying, first of all, and this is very similar to what HubMap is doing now, there was a notion of so I'm going to actually skip to the second bullet point here, a notion of a digital snapshot, saying that for a given tissue, whether it's a simulated tissue or real tissue, you're going to have some spatial structure, and you could segment it and find all the different cells. You might be able to identify all the different cell types and spatially annotate those. And then you're also going to have a characterization of the environment, things like what are the diffusible chemical species here? What's the matrix? What's the density of a cellular matrix? What's the composition of the matrix? Is it isotropic or anisotropic? What's the mean fiber alignment as you move across the tissue? Or you might characterize, say, the positions of the blood vessels and the radius and where they might form branch and node points in a vascular network topology. And so these are things that you could kind of objectively get by, say, cell segmentation or vascular segmentation and reading out that you might want to record. And the neat thing is, if you can agree on the characterization, then you can start developing tools that can visualize both experimental data, clinical data, and simulation data. And so we want to kind of facilitate just a description of a single snapshot in time of some spatial tissue, whether, again, was a simulation tissue or another tissue. This is where HubMap, I think, is getting very, very close now, because we have these tissue segments that can be mapped. They can be sliced and registered into a coordinate system. And then with that tissue, you can go and identify all the cells and identify their positions. And you can kind of keep that registry, say, here's a cell at XYZ, it has the following type, and then here is the original saRNA-seq data that maps to it. We're not quite there at that characterization yet. We're still kind of at here's a tissue and it has a bag of cells with the following population distribution, but we're getting closer and closer to that single cell, spatial transcriptomics type of characterization. And so that's something we envisioned, years ago, and certainly for simulation models where you have perfect knowledge, you can definitely write all that down. And the experimental side and HubMap is now catching up. On the flip side, say, now that I've identified all these different cell types, we need a notion of a digital cell line that says, what are the rules of that cell type? And so we decided to structure those rules first around by just a pure characterization of the key processes, realizing that we might be able to think of that as a phenotype and come up with some objective measures. So let's talk a little bit more about digital cell lines, because that's really where we've been focused for the last several years here in our simulation modeling. And so a digital cell line, like any data model, is going to have a combination of metadata and then actual measurements. And so at the time, we wanted to couple, saying that anytime we measure phenotype, cell behavior in some way, it has a very specific context. Because if you take that same cell and move it to a different condition, it's going to have completely different behavioral characteristics. And so we, said that being an observational data set, that a digital cell line really needs to say that I'm going to take some set of conditions, say hypoxia with normal glucose conditions, and I'm going to measure, say, cell motility, cell cycling, cell death, and add up all these things and just annotate it as well as I can. And then I take that same cell line and I'm going to go plate it in new conditions, say, and hyperoxia or physioxia, say 38 millimeters partial pressure of oxygen, and maybe serum starved conditions, and go measure it again. Then maybe measure it one more time and say, now put in doxorubicin and measure the properties again, or put in a growth factor, measure it again. But for every set of measurements, you need to capture that microenvironmental context for that data to be meaningful and useful. And in particular, you need to structure the measurements in a way that can be extended as you come up with new ways of measurement, but as a way that you can kind of get your head around and actually systemize it. And so what we decided to do is to define phenotype to be a behavioral phenotype, saying, that there's a lot that you can measure molecularly about a cell, but at the end of the day, there's a much more concrete set of things that the cells do. They can cycle, they can die, they can stick to each other, they can push each other around, they can crawl, they can secrete chemical factors or absorb and consume and sample them. They have some sizes and shapes. And now in terms of immunology, they have some immune interactions like phagocytosing other cells or attacking other cells and causing damage to them. Or as we're finding both in cancer and in virology, cells can actually fuse together and form multinucleate things. So we need to be able to characterize kind of these key processes and group them really by cell process as a way that gives you a method to systemize what you're measuring. But once you have a systemized way of representing cell behaviors, that also lends itself well to simulation and modeling, because now you can focus on implementing at least a reference model of all of those key processes. And any simulation framework can come up with their own reference model as long as it at least matches some of the key parameters that are measured, like how long should a cell cycle be? What's the death rate of a cell? How fast is it crawling? I mean, these are things that are fairly objective to measure, and you can have your own way of writing a mathematical model for how to implement those behaviors. But at least as you kind of hit those common denominator at the right time scales and spatial scales, there should be some agreement between the models. So we set out to start building several years ago a virtual laboratory that would allow us to probe these types of questions we want to ask about tissues and about how they're formed, how they maintain homeostasis, how they're imbalanced, how they respond to perturbations, how you model cancer, how it affects a tissue, and how a tissue might affect a cancer, and how all of those relationships may affect response to a treatment. And so we knew that we'd have to be able to model both the chemical environment in terms of different growth substrates, metabolites, signaling factors, drugs, and therapeutic compounds, and then the cells within those environments, where we know the environment, their behavior is context dependent. So kind of have like a molecular scale logic. We know they're going to be interacting mechanically, and those cells may have on an individual basis their own individual states and parameter values. And so that has driven us to something called agent-based modeling. So first we start off modeling the chemical environment. So we built a finite volume code called BioFEM that simulates a whole vector of diffusion equations. So you have like oxygen, glucose, a couple of growth factors, a couple of metabolites, and a couple of therapeutic compounds. you're very easily in the space of simulating 5 to 10 diffusing chemical substrates in 2D or in 3D, and you need to solve those simultaneously as you evolve your simulations. So that can be very computationally demanding. So we developed a parallelized method that works well across platforms, and notably, beyond just the standard reaction diffusion equations, we added point sources and sinks for individual cells. They can either secrete things into the environment or they can kind of consume them. And so that part was pretty much just the environment side. Then on top of that, we built something called PhysiCell, which is our agent-based model. Apparently this movie isn't working, so I will describe it instead. But the idea is that you have a collection of cells. So those point sources and sinks in the diffusion equation, you layer on now all the biological processes that we had talked about in our data model for cell phenotype. We have standardized cell cycle models that cells progress through different cell cycle phases with rate parameters given by the measurements in our cell cycle data model. We have a couple of death models, like in particular apoptotic death and necrotic death, and now there are just a bewildering variety of death models in virology and immunology. But really, at the end of the day, you want to model, say, how long does a cell live? And then once it's in the death process, how long does it take for that cell to kind of go through that death process? And does it release things in the environment that might affect other cells? Likewise, we've gone through that hierarchical phenotype, that data model, which kind of gave us a way to systematically, one by one, implement a reference version of each of those phenotype elements, so that computational modeling for us now becomes a matter of programming that phenotype, saying, because we have all these key processes worked out and it's parallelized and it works pretty efficiently, now the end users can say, I'm not going to worry about running really raw level code to say, how do I grow the cell volume? How do I split into two? Where do I put my daughter cells? How do I fix that segmentation fault because I broke something? And so that they can really focus more upon their biological hypotheses, saying that I observed that a cell in hypoxic conditions may increase secretion of vascular endothelial growth factor that diffuses into the environment, and maybe they upregulate cell motility. So they can say that I want to read these chemical signals and modulate the cell behavior. And really for them then becomes a matter of just increasing or decreasing parameter values that run these always on processes. And so it becomes a kind of a different paradigm to think about in the way you implement your models. So we published that a couple of years ago in PLOS Computational Biology. Now, let me kind of give you an example of taking this data model, right, this hierarchical structuring of cell phenotype, of cell behavior, and implementing those key behaviors, and then saying, now we're going to step back and say, we're going to introduce a broad variety of cell types. Each of them are going to have their own rules of how they respond to signals in their environment, which can include contact with other cell types to modulate their behaviors. And that forms complex systems. So several years ago, we formed a coalition, and we were quite fortunate to meet a really broad group of very motivated and wonderful scientists across immunology, biology, of course, computational mathematical modeling, to start building a comprehensive tissue simulator for COVID-19. The idea being that we would do iterative prototyping, we kind of build our current hypotheses, run it, simulate it, see what it does, talk to our team of experts and say what works, what doesn't work, set the next priorities for the next version, say, we're going to do these new hypotheses that help fix the things that were broken in the last version, and keep going. And then, of course, at every step of the way, we're releasing a new preprint so we can do the open science and fully open communication so that other people can learn from us. And then we share everything as open source software. And so really, we started off from an overnight prototype in March of 2020, then started recruiting in the community, and then gradually kind of stepped back from our role of being implementers of the models to being really more of the support team and the integrative team, and that different subgroups developed the key components of the model, and then we kind of helped to make sure that they were all compatible. And then gradually adding in more and more complexity to start more comprehensively modeling both the innate and adaptive immune responses, adding things like interferon signaling, the pro-inflammatory markers, macrophage polarization, immune cell trafficking between the locally infected tissue and the lymph nodes, and then T cell expansion and antibody production in the lymph nodes coming back into the tissues. And so we've been quite fortunate to work with a large coalition of over 40 collaborators from 20 some institutions, building this overall structure of a kind of a spatially resolved infected tissue, infected epithelial cells that have viral entry by ACE2 receptor trafficking, have a viral load in the individual cells, and then release a virus into the tissue. interacting with the local innate immune cells that will ramp up inflammation, and then demand trafficking to an ODE model of the lymphatic system. And then that will then have T cells and other cells come back into the tissue to drive the immune response. And so all these, and then, and then as we go, really, again, this kind of notion of building hypotheses that help you build the model. So now macrophages, by the version 5 of this model, I mean, there's a set of 12 hypotheses for macrophage behavior. And each of these hypotheses, right now, unfortunately, fairly laboriously become hand-coded as C++ functions in this computational framework. So we try to provide, a very clear API of saying how you modulate cell behavior, and then it's up to the modelers to kind of say, here are my signals, here are my hypotheses, and I'm going to create computational code that implements those hypotheses. So it enables very sophisticated work, but it does take a lot of manual work. And so, but here's kind of the notion of a model growing iteratively in complexity. So the first prototype says you start off with this blue sheet of epithelium. So cells go from blue to yellow as they get more and more infected. Single virus particle lands, it gets infected. It starts creating more virus particles that are released, diffuse into the environment, and then infect neighboring cells. So you can see these expanding regions of cell infection. And then black cells are the cells that die from the viral load. Then the version 2 of the model, we added more formally the receptor trafficking. So we added on more hypotheses, and the biology got a little bit more realistic. By the third version, this is where the community really started to take off and take over. They added notions of resident macrophages that would find infected cells, eat them, move into an M1 pro-inflammatory state, secrete pro-inflammatory factors that eventually start recruiting in other cell types. In particular, these pink ones are the activated macrophages, cyan is neutrophils, and the red ones are the CD8 positive T cells, the effector T cells that are attacking and killing off infected cells. And so now we're currently up to version 5 of the model, where you can start off with a naive tissue with some resident immune cells of a broad variety of types. And now some virus particles land in the tissue. And you can see that infection is definitely killing off a lot of tissue. But then as the immune system ramps up, the tissue damage slows down until you kind of end up at a steady state where you've resolved the infection. And here's the neat part. After you run this model and say, now take the immune cells and move them over to a previously uninfected tissue, and now you get a new infection. So let's say now this patient has kind of been exposed, they've developed their adaptive immunity, you still have all these antibodies floating around, you have a T cell population, and now when the immune cells land, it's protective. That this, there's a little bit of initial death of those first few infected cells, but the immune system did its job, and you don't have that huge tissue damage the second time around. And so this gives you the idea, right, that a basic, system, a framework where you have individual cells and that can model signal-based behaviors on the individual cells. You have hypotheses of this cell responds to this signal to do that behavior. When you start adding that up and have a community together to write down these hypotheses and implement them, you can arrive at some pretty sophisticated biology. But there's a lot to go, right? We've run into problems, right? That every round of our iterative modeling has gotten slower and slower and slower. The first iteration took 12 hours. Second iteration took a week. Third iteration took a couple of months. The fourth and fifth iteration have taken on the order of 6 to 12 months, because as the model grows and these hand-coded models grow, the complexity grows too. And it gets harder for this group to understand the full model. It gets harder for us to clearly communicate the current biological hypotheses of our model, which makes it harder for domain experts to participate in real time and provide that quality control that we know that we need. So our goal now is to say, can we learn from this process and do it again? Can we create a formalism that we can now write the hypotheses in plain English so that biologists and domain experts can participate in real time, say, I'm going to write down my hypotheses in this controlled vocabulary of some kind. And then if we can translate those hypotheses directly into mathematics and code, that means that we can get real-time collaboration with our domain experts. They can give us a hypothesis and we can run it right away without recompiling code and see what it does. And if we can reach that ideal, then we can start turning mathematical modeling into just a process of knowledge mapping. And that's really where we come back to the hub map. The hub map has gathered the identities of all the cell types. And now we need to go talk to those domain experts and say, now let's capture the knowledge of what those cell types do. And if we put those two together, we can say, import a picture from the hub map, import the rules of what those cells do, and now we have what we need to do to model the dynamics of the tissue. And if we can accomplish that, we transform that static spatial map into a dynamical map that encapsulates saying, here's what we think we know about the biology today. And if we put that together in the picture, here's what it does. Either the tissue works the way we expect it to, fantastic, we think we know what we think we know, or it doesn't. We say, you know what, we're missing some hypothesis. It's time to go back and fix something and explore it. And the mathematical model gives you this opportunity. So I'd like to just kind of spend the last couple of minutes showing some progress we've made towards this new ideal. And this is really an exciting and kind of, for me, a really exciting area, because I've been trying to do this. I've been thinking about this for about 10 or 12 years now, and finally feel like we've reached the point of experience where we've run enough complex models to kind of find the general trends and build that unified vocabulary and formalism that I think is going to make this actually work. And so, taking a step back across all the cancer projects we've done, the immunology projects we've done, the virology projects we've done, the digital twins projects we've done, we really do see a fairly common trend that a lot of the knowledge turns in a knowledge map is going to look like signaling factor X increases cell birth in cell type Y, or contact with some cell type increases death in some other cell type, or some cell type will eat another cell type upon contact, or pressure reduces some behavior. But really, if you abstract this out, all these are really statements saying that the cell has a behavioral response to a signal in the environment. And so, first we want to be able to kind of make a human readable language saying that for some cell type, I want to write statements that have a grammar like this, that some signal increases or decreases a behavior with some parameter values. And signals can be things like extracellular chemical concentrations, like say oxygen or glucose. It might be an intracellular chemical concentration, could be your pressure, contact with a specific cell type. And then the behaviors can be things like cell cycle entry or adhesion or an apoptotic death rate or rate of attack for a specific cell type. Or type changes like differentiation or transdifferentiation. And so really we want to be able to structure rules like macrophage says a phagocytosis tumor cell will increase secretion of IL-6, a pro-inflammatory factor, or CD8 positive T cell upon contact with a tumor cell may increase its lytic granule and perforin secretion rate, which has a very local diffusion rate. So basically on contact, it's going to start doing something. And so if we have a controlled vocabulary and a canonical representation, this data model lends itself to direct translation of these plain English language hypothesis statements and observations basically into mathematical structures that, for any one behavior, any behavior ultimately relates to a parameter value, because we built our model framework to say that phenotype is a hierarchical organization of key cell behaviors and processes, and each of those have key parameters to describe the rates. And so for any particular behavior, it's regulated by a parameter value. And so that parameter value can either go up or go down. And so let U be our total promoting signal, something that increases our behavior, D be the total inhibiting signal, something that decreases behavior. And we want to be able to say how changes in the total up and down signals modulate that parameter value. So promoters ought to increase their parameter value towards a maximum promoted value. So PU would be the maximum response under promoters. PD would be the maximum response under inhibitors. And then you have some kind of response function that maps it. And so typical response functions come straight out of pharmacodynamics, like say Hill response functions, or a lot of mathematical models tend to use simpler linear response functions. But as long as it's some monotonically non-decreasing function that goes between 0 and 1, we're home free. And so as an example, you can take the simple biological statement, interferon G promotes cell cycling. That becomes a mathematical rule. This signal increases this behavior, cell cycle entry, and we're going to use a Hill response function. Let's just say a Hill power of 1.5. Half max of 0.1, we have a base parameter value in the absence of signal, and let's suppose then our rules that it increases cell cycle entry tenfold. Well, the mathematical form is very easy to parse out of this statement if you have a canonical form. You have your modulated parameter value is your base rate plus your response function modulating you towards your maximum value. So notice that as interferon goes towards infinity, this factor goes towards 1 because it's a Hill function, and you end up at your maximum value of 10 R naught. And you get these beautiful plots that really show a clear relationship between, this qualitative model that we wrote out, this behavioral statement, and the mathematical form, and then the actual model behavior. And so T cell killing is an interesting one because actually a lot of things that we are used to seeing are actually compositions of simpler rules. Like CD8 positive T cells killing nearby tumor cells is actually 3 hypotheses altogether. The tumor cell contact with certain, with the recognized cell type would increase secretion of lytic granules. There's one, and so you have some kind of an increase in the secretion rate of that particular substrate. Then you might say that lytic granules, there's another rule for epithelial cells that say that upon contact with a granule, you're going to increase some kind of a damage parameter based upon a really simplified model. And then you might have 1/3 hypothesis saying that your apoptotic death rate, or whatever death rate you want to call it, increases with your damage. And so you just have another Hill response function. Put these 3 things together and you have basically T cell killing with very simple statements. So we've actually, written a prototype of this called the 3 Types app. We aren't going to really have time to live demo it in like a minute and a half, but I want to show that just even 3 cell types with some very basic rules where we have 4 signals secrete, A cells secrete A, B cells secrete B, C cells secrete C, and then there's a resource in the environment. And so we have 4 signals. We have A, B, and C, and resource, like say oxygen, and that we can modulate based upon these 4 signals the following behaviors: cell cycle entry, cell death, motility, and secretion. So really very simple, right? But here's what we can do. Say, let's suppose the cell type A is tumor cells, and they just secrete, like, say, tumor antigens, just shedding molecules into the environment, and that they consume resource, and that resource correlates with cell cycle entry. So there's the signals resource, increases cell cycle entry, and then resource, say oxygen, inhibits necrotic death. And then we'll get signal C in a moment. Cell type B are going to be some kind of like macrophages for us, but we're going to kind of concatenate some roles, saying that they have no birth and death for now, just keep it simple, that they are chemotaxing towards signal A, towards this tumor antigen, and upon contact, they're going to secrete, so in higher levels of A, so in regions of tumor, they're going to start secreting their signal B. Think of that as like a pro-inflammatory marker. And then effector cells, C, are going to say no birth and death, that they migrate towards signal B, so towards the inflammatory signal, and that when they reach a region of high inflammatory signal, they're going to secrete factor C, which is going to be like our lytic granules. And lytic granules promote death in the tumor cells. So very simple, right? We just take this system and write it down to a series of very simple statements and put it together with this signal response type of formulation. And you can play with this online. I'd be happy to walk you through it, one-on-one later. But you put this together, and within 5 minutes, you can basically build a very basic adaptive immune response. So here we have our tumor growing. These bright dots are dead tumor cells. And you see that the green cells, the macrophages, are kind of aggregating in regions of high tumor cell concentration, then drawing the blue effector cells towards them. And as they get high, they release their toxic cargo. They'll start killing off the tumor cells, which then becomes a region of low tumor cell concentrations. So the macrophages go somewhere else and then eventually draw the effector cells towards them. So you can see that a very basic markup, even a very basic markup, signals, modulating responses can start building up some and reproducing some very interesting, dynamical behaviors that start to get closer and closer to what our notion of what immunology should be doing. So ongoing work is, we're building ways to work with the hub map to import those static snapshots to say, here are the initial positions in the FTU and who is here, what are the different cell types and where are they? We're now working on this domain capture, saying if we can come up with this nice way to represent the knowledge and to map it onto mathematical models, we can say, here's the tissue, and for these identified cell types, here are the rules, and we can set it forward and have this dynamical model. And ultimately, we're trying to get this way that we can kind of create community efforts to start harvesting that domain knowledge and annotating to a curated resource where eventually we're going to accumulate enough knowledge to record what's going on, but make it completely reusable and implement it. So that's where we are today and kind of how that relates to the work in the hub map. And just wanted to point out that we do have an annual virtual workshop for PhysiCell for this modeling framework. And our next one is set for July 24th through 30th this summer, be fully virtual. There will be an application process for, so the tutorial sessions are completely open to the public. Anyone can dial in on Zoom. And then in the afternoons, we have a more selective group for a mentored hackathon to kind of build tools and build models using PhysiCell. And this year, we would expect to start doing a little bit more work with the HubMap data, and in particular, this knowledge mapping framework that we're developing now. So we'd be really delighted to see you there this summer. So I know we're really right up at the end of time, but I'd be delighted to take questions if you have them.
Thank you very much, Paul. This was absolutely fantastic. I'll just see if there are any questions from the audience. Anybody? Otherwise, I have a few.
I have a question.
Sure.
Can you hear me?
Yes, I can. Thank you.
Yeah. First, I really wanted to appreciate your effort in translating colloquial assumptions we usually have about biological systems into quantitative statements that can, inform our models. I think that's amazing, and the fact that you've engaged the community, that's like, it's some work that's like praisable in many directions. So first, I want to say that. The second is like, I think that usually in these kind of efforts, people, so I know this is not my field, but something I hear a lot is timescale integration. Like, how do you, for example, sometimes like the assumption that like the T cells produce granules at a certain speed might not be what you actually know. You actually know that there's this phosphorylation event that makes a certain molecule more permeable. And from there, you want to translate it into maybe the granules accumulating faster, something like that. Is this something that could be somehow incorporated into the model, or is this beyond the scope because things tend to be at a similar time scale?
Those are fantastic questions. So first of all, thank you very much for your kind words in the first part there. The second part is, there is always a choice, I think, in how sophisticated you want to make your model and at what level of mechanistic detail you want to get down to. Right now, I think, certainly there are ways to add more and more molecular detail to get to the molecular mechanism, right? To say, you have these little granules, but what they really do, they're digesting bits of cell membrane, and then that disruption of the membrane is affecting things in the mitochondria that are eventually going to, affect caspase responses, and it might affect death. And that's just one method of death, right? There are also activation of death receptors that can happen. So there's a lot that can happen in which to decide, what's the minimal set of rules that it takes to get there. And that often depends upon what you're trying to do. Right now, we're at a stage of cancer immunology and tumor immunology and just regular immunology, where even the rules of interaction between different cell types is not 100% crystal clear. We think we know a lot, right? And but clearly, by the fact that we can't seem to resolve autoimmune diseases, we can't quite always get cancer immunotherapy to work. We can't get tissues always to be accepted in say, transplants, means that we don't know as much as we think we do. And there are a lot of blind spots. And so, at that course level, we'd have to say, regardless of the molecular how of the interaction, we can kind of take a step back to say, what are the interactions? The two cells touch each other, that one recognizes the other and tries to kill it and induces death in it by whatever mechanism that might be. And for the sake of understanding the dynamics, the balance of those interactions, that course level description is probably good enough. Now, at some point, when you get down to pharmacologic intervention, you say, now I want to interfere with that interaction in some way. I want some kind of a drug, for example, that diffuses the environment and might inhibit that process or promote that process. Now that level of molecular mechanism becomes really, really important, right? So kind of underneath the wiring diagram of this interaction, there's a whole lot of molecular biology going on. And that's, I think, at that point, we really want to interfere in a very specific quantitative way. That becomes really important. So I think it kind of depends where you are on that stage of understanding this dynamical system and then getting to a point of very specifically interfering in that dynamical system. There are ways, however, to integrate that. One thing that I've been really excited about in my group, in particular, Randy Highland, who I think is on this call, has been working very closely with another student in my group named Furkan Kurtoglu to implement intracellular models. And so we've been very fortunate. Actually, another group we collaborate with is at Institute Curie, and they have developed something called MABAS, which is a networks of Boolean models. And so this kind of idea that you might have a gene network, for example, and maybe you can't fully parameterize it, but just even the network diagram structure itself forms a nice directed graph that becomes a Boolean network. And so thanks to their help the last few years, we have a capability to say in an individual cell line, digital cell line in our case, right? You can integrate a Boolean signaling network that helps you relate those chemical responses to downstream, intracellular processes to affect all the modular molecular behavioral parameters in the model. And if that's not good enough, if you really need true dynamics, now we have also been able to work with the Live Roadrunner team to say, maybe you really want to model the actual molecular dynamics of, say, the phosphorylation events that lead to this recognition of the cell interactions, driving some response. Well, we can actually write those as chemical reaction statements in systems biology markup language, and we can integrate an SPML model into an individual cell agent, run that model behaviors. Again, the neat thing is we have this nice glue between the two different codes that the cell agent is kind of responsible for interaction with the environment. So the cell agent samples the chemical substrates in the state and can feed that information to an SPML model. The SPML model turns around and does all the proper mechanistic biology to say, what are my new rate parameters, and writes those rate parameters back to the cell to modulate behavior. So we can actually add that mechanistic scale. But as you can imagine, it gets a lot computationally slower the more you do that. So you kind of, it's good to wait as long as you can to do that until you really truly need it. But at some point, I think you do need to do it, especially if you're getting into quantitative pharmacology.
Yeah, just to follow up to that, I think that is like a well-guided effort. Like, I think often the way to merge these timescales is to ask, like, how can this low-level thing inform how we think about this high-level thing? Like, for example, you're talking about pharmacodynamics of a particular drug, and probably like what you want to do is like interfere with this, I don't know, receptors expression or some like high level. So kind of like maybe borrow from another model, like how much can this drug actually affect this rate that I use in my high level model? And that way kind of like get from the drug to the intended effect to the overall effect on the more complex system.
That's a beautiful point and a good comment, because we talked a lot about the multiple spatial scales, right? That you have the cells, they all have their individual states that kind of work together to kind of drive the tissue level dynamics. But there's also the multiple time scales, like you mentioned. There's the molecular time scale, which is super fast, mechanics time scale, which is kind of intermediate, and the cell behaviors, which plays out over the order of minutes, hours, and days. So there's this huge separation of time scales, and you use that to your advantage. In fact, in PhysiCell, we do that. We actually have 3 delta T's in our code right now. In fact, now 4 delta T's. That we have the intracellular time scale, we have the diffusion time scale, we have the cell mechanics time scale, and we have the cell phenotype time scale. And the way we've written the code is that we don't have to do everything at every time step. Now, some things are fast and just have to do it at every time. So tick, 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 do 10 diffusion steps, do a mechanics step. Another 10 diffusion steps, do a mechanics step. Do that 600 times, there's a phenotype step, right? Some fast processes are fast, some are slow. But there are other things you can do kind of more analytically, right? Imagine say the Michaelis-Menten type dynamics, that lead you to a Hill function really come down from the fact that, say, like you have a catalyst and an enzyme or something, that often some of the reactions are really fast compared to the other ones that become rate limiting. And so you end up with these saturation curves, right? And so when you take a step back and abstract, generalize that a lot of mathematical models really involve saying what happens fast, what happens slow, what at the perspective of the fast scale is fixed because it's moving so slowly that it might as well be a fixed boundary condition. And so for molecular dynamics, you can do a lot of that, right? Say, the cell might as well be a fixed static thing compared to molecular dynamics of, say, phosphorylation. On the other hand, as far as the cell goes, phosphorylation is happening so fast, it's like quasi-equilibrium, saying that the state of the molecular model is basically a function of my current geometry and other properties. And so that separation of timescales can be really helpful, because what you can do is say, take your molecular scale models and study them offline in high throughput, and understand the kind of, that becomes a constitutive relation to put into the higher scale model. And that kind of separation of scales can be really beneficial, right? You end up building a hypothesis that some signaling factor varies with some behavioral value. That really is hiding, like you say, a lot of middle steps. Let's say, the signal leads down to this binding with the receptors, leads to this change in the protein interaction dynamics, which leads to a change in the transcription factor, which eventually leads to a change in the cell behavioral response, right? There's a lot going on in there, which leads to some lagginess in the system, which is also fascinating. And so, we've actually been running with that now. In fact, you have to. Like we've been doing cell metabolism models. And metabolism models, say if you're using a flux balance analysis model, is a fascinating thing. We're really lucky, again, also to be working with Barcelona Supercomputing Center on flux balance, with Miguel in particular, that at any fixed time step, basically flux balance analysis models are solving constrained optimization problems. So you say, for my cell in this particular state, with these extracellular substrates and these intracellular substrates and my rate parameters, I solve to steady state a constrained optimization problem to say what my new flux rates are for all my substrates, either in or out. And then I move a little bit of substrate into my cell, create energy, make biomass, and move on my merry way. That's one optimization solution per cell, per fast time step. Now I want to simulate 10 to the 6 cells, and I have 10 to the 2 time steps per simulated minute. That's a lot of optimization problems. There's just absolutely no way that's going to work. And so, yay, we can write the software interfaces, but we're waiting until, the sun burns out before we finish this thing. And so what we have to do is be smarter about it. So now we're starting to build surrogate models where you take, your very detailed molecular scale model, and you take advantage of a high throughput cluster. You say, I'm going to run my 10 to the 8 simulations across input space and measure all the outputs. Now I've got my features, my inputs, I've got my labels, my outputs, and I form a deep neural network and train it up offline. And now I'm going to build that as a circuit model for my molecular scale process, which is kind of like the deep learning version of this observation, right? Say, I have these inputs, I run my model in high throughput, explore all the possible outputs, and I form a hypothesis of kind of the functional form. And sometimes we don't come up with a hypothesis, right? It's just too complicated, too nonlinear. And that's where deep neural networks are beautiful, that we can kind of automagically capture these relationships. And so we're now building this pipeline to say, run your intracellular model, however complex it might be, in high throughput on a cluster, go mine the results, train a deep neural network, and then we're going to import that deep neural network into PhysiCell to directly map from the inputs to the outputs without running the intracellular scale problem all the time. And it's a work in progress. Preliminary results are promising, but we're still the jury's still out if it's going to work. But I feel cautiously optimistic that it will, because you have to make that tractable to get that mechanistic level of detail, but still be able to get somewhere with your larger scale simulations.
Thank you. That sounds very exciting.
Of course, theory can be way faster than deep learning. So whenever you have theory to drive it, that's I think always going to be preferable. These are fun questions.
Thank you.
Yeah.
Are there other questions? I know we've run a little bit over time, but if there are any other questions, I think now is the time.
If we have still a little bit of time, I would maybe ask one question. I would join Raphael and Nico and really thanking you, Paul, for a fascinating and great talk. I wanted to go back a little bit from the simulation to the first part of the talk where you talked about HubMap and these fascinating ASCT plus B tables. My only question, I managed to spell it out correctly. I'm wondering to which extent these tables keep on evolving, right? To which extent you're putting these tables out to the public. I could almost imagine some kind of a Wikipedia on top of it where the experts are keeping on discussing. You showed how much work there was involved in having this Zoom discussion. Is Paul still with us?
I think perhaps
I'm still with you, but you were chopping up, so turn my video camera off to save a little bandwidth. Okay. I'm in the hotel Wi-Fi, so
I see. Yeah, I was just asking about these ASCT plus B tables, to which extent they keep on evolving. given that I can expect, for example, these marker genes that we have for individual tissues. It's quite interesting when you talk to the experts. We talked with one lab here at HMS working at vascular endothelial cells. And it was quite interesting when you asked them, which are obviously the experts in the field, what are markers for this tissue, and how was this different to other groups working in the field? So I can imagine there's quite some controversy in the particular tissue for particular cell types, which each themselves are like subject of ongoing research. So a long question short, to which extent do these tables keep on evolving and are shared with the public?
I think that's a beautifully insightful question. I think they're absolutely going to keep evolving. You hope that they get more and more stable. The tuning is kind of at the edges, right? I mean, you don't want some fundamentally basic thing to keep changing, like, you know, what we call an epithelial cell today becomes an endothelial cell tomorrow. You know, that you hope that there's some stability in the things that we all agree on. But you're absolutely right. First of all, you know, getting any community of scientists to agree on anything is kind of an exercise in herding cats. And so I'm kind of in awe that Kadi Brunner and Ellen and the others have managed to get 24 of these things built and get community input and some reasonable domain consensus. That's already, to me, humongously impressive. And I'm just kind of in awe of that effort. And I know it's come down to a hell of a lot of phone calls. But at the end of the day, you know, you're right. These are, you know, knowledge isn't static, right? We keep discovering things, and our models of things change over time. I mean, I kind of think back to, you know, our general model of the atom, right? You know, and we went from basically thinking of atoms as plum pudding to what we have today with subatomic particles. You know, I mean, it evolved over time, and it got, you know, there's some things that kind of converged over time, but, you know, every now and then there are shakeups, and you have to be ready for that. And so what I do like about these ASCT tables, plus or minus B, is that it kind of forces us to sit down and say and write down what we think we know. And, you know, that's a snapshot of our community today. That's a really valuable thing, because how often, you know, how much do we go through our day-to-day work where we don't even write down what our operating assumptions are? There's kind of this, you know, moving, morphing thing. And that's, I think, the great opportunity of both data modeling and mathematical modeling. Is it's an opportunity and a requirement to sit down and write down what you think you know. And so then the key is to say that, you know, we know these tables are going to change over time. So it's going to be really critical that whenever you write something down that relies upon these objects, that we're going to have to write down the proper provenance metadata. Let's say, you know, I used my liver ASCT table version 3.7, you know, and numbered releases become really important, just like simulation modeling for reproducibility. We have been very adamant that you know, and some groups don't do this, right? But we've been very adamant that our development branch and our master branch have to be separate. And I don't want anyone doing modeling with the development branch, because I want people using very specific and clearly numbered releases to say, I use, you know, download version 3.1.7 or whatever it is. And that's what I used to build this model. So people understand what the current operating assumptions were at the time they built that thing. And we've been trying to do that with our COVID modeling too, right? You know, that we say, here's version 5 of the model, and we wrote down all the macrophage hypotheses, all the neutrophil hypotheses, and we wrote this out fairly laboriously. But the problem, right, of course, is that it's not automated. So it's hard to do that like with good quality control. So ultimately, for these things to work well, realizing that these are going to be morphing, evolving objects, is we have to make it easy for people to do the right thing. There have to be tools that help automate, you know, these annotations and the metadata, so that, you know, there's less human intervention to do it, and that over time, the process of writing down hypotheses and turning it into code becomes more and more automated, so that you don't have these interpretation issues. But, you know, it's definitely going to be a moving process, and to me, that's kind of the beauty of it, is that these things are going to evolve with the community and its understanding, and hopefully that understanding I hope the community continues to evolve, right? I hope these don't stay static. I mean, if it's done, great, then the science is done and it can become all engineering. You know, engineering is when we completely know what we're doing, and science is when we're finding out that the engineering doesn't work yet. So
Yeah.
I think it just reflects the fact that it's still science.
Yeah, I think these tables would likely benefit from, you know, some ways for collaborative developments, things like GitHub issues or commenting or these kind of things. I don't know whether this is feasible or not, but I think that would be great, because then experts that maybe not necessarily have been part of the Zoom call could go there themselves and like, you know, start a discussion on it, like via these commenting features.
That's a beautiful idea. And I think as we get into more of the knowledge mapping part, we'll probably be using something similar to that. I can't completely speak for Kali and the way that she's run it, but I know that she's had a very engaging, you know, community engaged method where they run a lot of workshops to get these domain experts into a room together to kind of write out what they think they know and to come to consensus on these. And so there is a process of community feedback and refinement. Now, could that be turned more Wikipedia-like? I think it could be, and I think it's a nice idea. Of course, you know, the more open something is, the more chaotic it is, right? And it's difficult to understand where you are. So it takes a fine balance, I think, between being completely flat structure, you know, democratic. No one is, you know, no one is in charge, which means that no one is responsible either, right? And that could be, you know, and then no one's accountable in a structure that's completely flat. Like a human structure, right? That's flat. And so you need kind of you do need some people who are accountable for curation and ultimately saying, you know, we've taken all the input and here's what we're going to put out. Because at some point you have to make decisions. Otherwise nothing gets done. And so I do think they found a nice balance on that. But there are a lot of great tools and perspectives, I think, that can help facilitate that automated, you know, issue reporting, for example, say, hey, this particular node in my knowledge map, I disagree with, right? And kind of raise an issue. I think that'd be a really cool thing. As long as it doesn't get basically denial of service by too many submissions. And so, you know, I think it's, you know, ultimately that gets down to the sociology of science. And I think it's just exhilarating.
Thank you.
Excellent. Well, I think that we've had a few minutes of discussion and a wonderful presentation. Thank you again, Paul. I'm going to ask for a round of applause, however people wanting to do that, and thank Paul for this wonderful talk and to close off the seminar now.
Well, I really, again, appreciate the invitation and the wonderful discussion with all of you, and I'd be delighted to follow up and just have another virtual lab visit and discussion.
Certainly. If you want to hang on the line a few minutes, maybe we can already talk a little bit and schedule something.
Yeah, although I have to, my family's
okay.
Actually, I probably better go because my family's in a museum that's more than a half-hour walk away that closes in a half an hour. So like I'm
for sure
abandoning my family in New York City. So I need to
go to
them in a minute.
Certainly.
But I'm delighted to follow up by email and schedule a follow-up chat. Thank you so much. This has
Absolutely.
been a really fun time.
Yeah, it was fun. Thank you. And I'll follow up by email then.
Thank you. Again, thanks for the invitation. I greatly appreciate it.
Certainly. All right. Thank you.
Thank you.
Have a good afternoon, everyone. Bye.
Thanks, Paul. Thanks, Rafa.